Okay.
Yeah.
Okay. Next is Matteo Fabbri from the Field Museum.
Okay. Okay. Thank you, everyone, for having me here. It's my privilege to present you some of my research that is going on and will start in summer in my lab at Johns Hopkins. Obviously, I don't have to explain to you why phenotype, what it is, and why we like it. But maybe I can remind you why structure and morphology is so important. And the reason is that structure is the only thing that connects the history of life, extinct and modern. Molecular work can go just a few million of years, therefore genomes and molecular data are only restricted to more recent species. Therefore, morphology is the only way we can bridge the history of life, where the fossil record is the representation of the majority of diversity that ever lived on our planet. And therefore, it's fundamental to understand the origin of clades and to date the origin of clades and also to figure out where these clades actually appear. But on the other hand, the fossil record is incredibly difficult to interpret unless we understand what's the connection between morphology, ecology, behavior, and so on and so forth using modern species. So in other words, the fossil record and modern diversity are complementary faces of the same coin that depict the history of life. And we can basically put together these sources of data through quantitative comparative methods and 3D imaging. This is because not only we can extrapolate information from the three-dimensionality of the structure, but we can characterize this and put together these sources of data because the structure has an evolutionary history that makes structure homologous. But we have a problem regarding the modern species because until now, major efforts have been done on adult phenotypes, trying to capture the evolutionary diversity of life. But there is an incredibly important resource of data from extant species that is still way behind, and I would say the most behind source of data in terms of collection, and this is developmental biology. In other words, embryos have been fundamental to understand how morphology arises generationally, but it also follows specific roles that have been the scaffold for the history of life since its beginnings. And the general reason for that is because we can actually track morphological change through development, starting from the phylotypic stage, which is this moment in organogenesis where morphology is extremely conserved, and only later on in development, the clade and taxon-specific traits that characterize modern diversity is actually arising. So this means that if we put together an evolutionary framework that includes the early moments of development, we can actually bridge evolutionary biology with developmental biology. And from a molecular perspective, we are doing spectacular because we had the vertebrate genome project, one genome sequenced for every single vertebrate alive today. Spatial transcriptomics is bridging gene expression with morphology. And CRISPR-Cas is the experimental attitude of twitching the genome to understand how a different genome can bring different phenotypes. But when we think about phenotype per se, we are far behind, and this is mainly the kind of leftover of the tradition between the 50s and the 90s, where Alchan blue and alizarin red is the main way to quantify and show skeletogenesis. The problem of this technique is that all the soft tissues are completely erased, And the skeleton per se is only one part of development of the organism per se. But there are different ways, as you all know, that we can use. So 8 years ago, I decided to go through the museum collections and looking for non-model organisms that can actually let developmental biology move over from the 5 model organisms we use since Aristoteles. And the museum collections are the only place where non-model organisms are actually created, because due to the collection of endangered species and species that are actually today extinct, in your collections, you have embryos of animals that are completely impossible to sample today. And therefore, I decided to go in the collections of fluids for bats, for primates, as in this case. And it's insane to see how samples like these Ahelobates, still completely nutritious, has here a full embryo in complete development. And this kind of samples is not only telling us about developmental biology, it's telling us, for example, how the evolution of the interaction between adult and embryo evolved in primates, including our species. We can also go towards a synchrotron, and I hope this works. So the synchrotron for smaller samples, this doesn't require staining, but allow us to go to subcellular resolution in terms of embryonic samples. And in case we are lucky enough to get fresh samples, I know that some of your people in the museums are going around for fieldwork to get new samples. Sometimes the females are pregnant. In case of this kind of fresh sample, we can use a technique that was invented in 2013 in order to image a structure of the brain. And in this case, this technique takes out all the lipids from the sample, and then we can use primary antibody and fluorescent antibodies in order to image in 3D the early developing structures of the skeleton. And this is one of the examples where we have a single embryo, but I can put multiple antibodies targeting different systems and image this in 3D. And this can be done across one developmental series or multiple developmental series in order to track what are the small changes early in development that are responsible for the extreme phenotype of the adult. And this is an example, very qualitative, but here we have a shark, and here we have a skate. And this is one of the early moments in development where we are starting to see this divergence in the phenotype. And this is how it looks later on in development. So our skate that has here the pectoral fin, like any other chondrichthyans, grows towards the extreme phenotype. Here I just want to show you how you can image this at the cellular level. So we are talking about centimeters long samples that are cellular level accuracy. And so if we do this, including CT scan, synchrotron, and clarity and immunofluorescence across the tree of life, at this point, we can actually infer how development influences evolution from a mechanistic level, which is something that is not possible just looking at modern diversity and the fossil record, just because we have to understand what are the constraints in development that dictate the rules of phenotype. And at that point, we can infer why different phenotypes can indeed appear. And so in the last 8 years, I collected almost 900 embryonic samples that belong to 43 different species, spanning from chondrichthyans to humans. But obviously, this is just a proof of concept. And so what I'm trying to do today is to open a call to all of you, saying that if you go in your fluid collection and you find embryonic samples, I would be more than glad to collaborate and figure out how we can expand our sampling within the tree of life. And as a plus, all these scans, independently from the method that you are using, are three-dimensional. So what these data mean translated is that we can actually investigate the history of life in 5 dimensions. 2 are time, development and evolution, and 3 are space, the morphology per se. And so with this, I want to thank all my collaborators and fund resources, and thank you for your attention.